Hi everyone, this is lecture 4, part 2, eukaryotic cells. So from this point on, we're going to talk mostly about eukaryotic cells. And so eukaryotic cells have internal membranes that divide the cell into compartments, the organelles. And so organelle literally means tiny organ. So cute. And if you think about, how one cell can be a whole organism, it really is kind of like a good analogy to our organs, how each organelle has like a separate space to do different things. So the cell's compartments provide different local environments so that incompatible processes can occur in a single cell. So for example, like in your stomach is a very acidic environment. There's a lot of acids and enzymes to break things down. You want that to be totally separate from like your heart that pumps the blood that's going to go to every cell in your body, right? So having different compartments to have different local environments is pretty important. And so that's how organelles kind of are for the cell. There's tiny little different local environments that can have incompatible processes occur in a single cell. And so the basic fabric of biological membranes is a double layer of phospholipids and other lipids. And as we go through all these different organelles, it's going to be the same for a lot of like plant and animal cells. Plant and animal cells have most of the same organelles, and so when we get to the ones that are specific to just plant cells, I'll point it out, but they have most of the same organelles. So this is kind of like a generic, typical eukaryotic cell. So there is a plasma membrane that all cells have. There is the cytosol, that like just fluid goop. There is DNA housed inside of a nucleus in a eukaryotic cell. And then there are ribosomes that are very, very tiny, so you can hardly see them here. But we'll talk about those. And even though we kind of show a model of a generic cell, not all cells look like this. So especially in different organisms and in your body even though we have eukaryotic cells, we have cells that might be specialized for specific purposes. So they might have more or less of certain organelles based on what its like purpose is in the body. So yeah, we teach about this generic cell, but not all cells look like this. Like the neurons that are in the, my background, they have the parts that are necessary to be cells, but they look a lot different than this kind of cell. And so, yeah, just to point that out. So the nucleus is information central. So I tried to have the headers with like the main just bullet point what it does to help. So the nucleus is information central. The nucleus contains most of the DNA in a eukaryotic cell. And then the ribosomes use the information from the DNA to make proteins. And so it's kind of hard to see in this picture. I kind of just, zoomed this picture and scaled it up, but the resolution isn't that good. Sorry. So the nucleus contains most of its DNA in a eukaryotic cell. And if you were to compare a cell also to like a city, people say that the nucleus is like the mayor's office, or like the main, city government office, city hall. So because it has like the important things that are gonna dictate everything else in the cell. The nucleus is also usually the most conspicuous organelle, meaning it's most like obvious to see. And we did see that in those pictures. In part 1 of this lecture, you could see, not that much detail, but you could see the nucleus in the middle. So it is a pretty large organelle compared to all the others. And it's the most like conspicuous. And the nuclear envelope is its own membrane that encases the nucleus. So the nuclear envelope encloses the nucleus and separates it from the cytoplasm and the rest of the cell. And it's kind of hard to see in this, again, low resolution picture, but the nuclear envelope is actually a double membrane. And so each membrane has the lipid bilayer, but there's actually two layers of membrane. So this picture is a little bit better actually in resolution. So this is the nucleus in purple. This is the nuclear envelope in kind of like a darker purple. And there are these little holes called pores, kind of like how the pores in our face are like little holes for stuff to get in or out of. And the nucleus is kind of the same. There are pores that control, whether something can go in or out of the nucleus. And it's not just a hole, it is a complex. There's the pore complex that makes the little pore. And then you can see there are also some ribosomes that are actually bound to the nuclear envelope. So the pores are aligned with a structure called a pore complex, and they regulate the entry and exit of molecules from the nucleus. And so on the inside, the nuclear side of the envelope is lined by what's called the nuclear lamina, which is made of proteins and maintains the shape of the nucleus. And there's also evidence for a nuclear matrix, so like a framework of protein fibers inside the nucleus that are throughout the interior of the nucleus to also, again, help maintain the shape. And notice how it says there's evidence for, so there are still some things that we're still learning about the cell. So if I come back to this picture, you see that pore complex, and that is what makes those little pore holes. So this is a picture of a nucleus, a TEM image, and so you can see the pores. And if we zoom in, they're not just little holes, they're these complexes that you see that make up those pores. And then on the inside is the nuclear lamina. And then also to point out, because I'm on this slide, there is DNA. So here, kind of small, this like blue string looking stuff, that's DNA. And DNA can be wrapped around proteins called histones. And so when the DNA is wrapped around the proteins, we refer to that as chromatin. And so usually chromatin, it's DNA around proteins, and it's kind of loose and it just hangs out in the nucleus. So inside the nucleus, DNA is organized into units called chromosomes, and each chromosome is one DNA molecule wrapped around proteins, and together they're called chromatin. And usually it's pretty loose, but chromatin condenses into what we think of as chromosomes and what we picture as chromosomes when a cell is preparing to divide. So normally chromatin is kind of loose like that, and then it only condenses when it's ready and preparing to divide. And also inside the nucleus is the nucleolus, which is the site of ribosomal RNA synthesis. So this darker purple spot is the nucleolus. So next up are ribosomes. Ribosomes are protein factories. So again, if you think of the cell as a city, the ribosomes are like a factory. It's like if Amazon just had a bunch of 3D printers and made everything there. So ribosomes are protein factories. They make proteins, specifically those macromolecules. And we tend to show them as kind of just these blobs, again. But they are actually complexes made of ribosomal RNA and protein. So not pictured like this. And so ribosomes can be free-floating in the cytosol, and so we would call those free ribosomes. #free ribosomes. or they can be stuck onto other organelles. So they could be stuck onto the outside of the endoplasmic reticulum, which we'll talk about in a couple slides, or on the nuclear envelope, as we saw. And so then these are referred to as bound ribosomes. They're bound to something. And so like I mentioned, we kind of show a little diagram of ribosomes as like these just blobs. And I like to say it kind of looks like a hamburger bun. There's like a larger top bun and then the smaller top or bottom bun. But again, that's just like a simplification, how we show it. And sometimes it's just like little dots. But realistically, it's part RNA and part protein. And as we covered, proteins can be huge and have really complex structures. So here's a computer model of a ribosome and all the different, layers and structures. And here's a TEM image of some free ribosomes in the cytosol, and then some that are bound to the endoplasmic reticulum. So there is a combination of organelles that work together in what's called the endomembrane system. And so that includes a couple things that we already covered, like the nuclear envelope and the plasma membrane. But it includes some other new things too. So I'll talk about these on the next few slides. But so all of these work together in this endomembrane system. And they are either continuous, like one flows into the next one, or they are connected via transfer by vesicles. So we're going to talk about all of these, starting with the endoplasmic reticulum, because we already covered the nuclear envelope. So the endoplasmic reticulum is also like a factory, but it makes other macromolecules. So it's a biosynthetic factory, and there's two parts of it actually. So here it is in this like light blue, and it's pretty large, right? It actually accounts for more than half of the total membrane in many eukaryotic cells. And it's kind of, again, hard to see in this figure, but it's actually continuous with the nuclear envelope. I think there's a better picture on the next slide. And there are two regions of the ER. There is the smooth ER. So if we were to look at it under a microscope, it looks more like smooth. As if you were to touch it, it would be smooth. And then the rough ER, like if you were to touch it, it's like it would be rough, because there are ribosomes stuck on. So it makes it like look more rough. So here's a picture with a better resolution. So the nuclear envelope, you could kind of see it like just is continuous with the endoplasmic reticulum in blue at some parts. And there's the smooth ER that looks a little bit more like coral, kind of, I think. And then there's the rough ER that is a little bit more like flattened, but it has those ribosomes like stuck on. And so the ER can like kind of bubble off, like pinch off and form this bubble called a transport vesicle. And that's kind of like a little, like a little transport pod to send stuff around. So here's a picture, I assume a TEM image also, of the smooth ER and the ribosomes that are bound to it. And then, or the rough ER, sorry. And then this is the smooth ER that kind of looks different and is a little bit more like coral looking. And so to talk about what each part does, so the smooth ER makes lipids. It also detoxifies drugs and poisons and stores calcium ions. So again, some cells in your body are going to be specialized to do certain things. And so some cells might have more smooth ER. For example, cells in the liver that are, intended to help detoxify stuff. Or cells that are involved with bones because they store calcium ions. So yeah, the smooth ER does some of its own stuff. And then the rough ER, because it has the ribosomes, it's part of like making proteins. So the rough ER has the bound ribosomes, and will secrete glycoproteins. So these are proteins that are covalently bonded to carbohydrates. So if you see the prefix glyco, that usually has to do with carbs. So glycoproteins have a carbohydrate stuck on. And these also secrete transport vesicles, and so they'll secrete proteins surrounded by these membranes and these little transport vesicles, kind of like a little transport vehicle. And it's also a membrane factory for the cell. So altogether, the endoplasmic reticulum biosynthetic factory makes certain macromolecules and then sends out the transport vesicles. So next up in the endomembrane system is the Golgi apparatus. And so this is the shipping and receiving center. So this is kind of like the UPS or USPS, whatever shipping company. And so the Golgi apparatus, I think looks really cool. It consists of flattened membranous sacs called cisternae. So it looks a little bit similar to the ER, but it's different. It's a lot more like flattened and obviously smooth, and it is detached from the ER. So stuff has to get sent to the Golgi apparatus through those transport vesicles. And so when it's there, the Golgi apparatus modifies products of the ER, makes certain macromolecules, and sorts and packages materials into transport vesicles, again, to get sent somewhere else. So it's kind of like if something was, made in the factory of Amazon, but then it gets sent to the post office, and maybe there's some modifications that have to get made before it gets shipped off. Yeah. So then it'll get packaged the right way and then shipped off to wherever it's going. So here's another picture showing that there is a receiving side and then a shipping side. So the cis face, usually cis means on the same side. So this is on the same side as like the ER. So this is the receiving side. A transport vesicle from there will come on this side. Whatever the products are that are made there might get modified here in the Golgi apparatus and then get sent off. So this is the shipping side. Trans face means different or opposite, and so the trans face is the shipping side that sends stuff out. And then here's a picture of the Golgi apparatus. Wide flattened sacs, and then the vesicles. So lysosomes are a kind of vesicle, pretty much. And it's not always necessary, but sometimes. So lysosomes are these digestive compartments. And so a lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. So if you remember hydrolysis, hydro means water, and then lysis means breaking. So hydrolytic enzymes break down macromolecules. So they'll be inside these sacs of lysosomes. And this environment is actually pretty acidic. So the lysosomal enzymes work best in the acidic environment inside the lysosome. So again, it's nice to have these membranes that separate these really acidic environments from the rest of the cell. And so these hydrolytic enzymes and the lysosomal membranes are made by the rough ER, and then transferred to the Golgi apparatus for further processing. And some lysosomes probably arise by budding from the trans face of the Golgi apparatus. So again, there's still some stuff that we're learning more about, and that we could say like probably arise by this way. And so some types of cells For example, some white blood cells can engulf another cell by this process called phagocytosis. So this cell will engulf either a whole nother cell or something from the environment in this process called phagocytosis, and this forms what's called a food vacuole. So these lysosomes can then fuse with that food vacuole and digest the contents. So that's how they're kind of like digestive compartments. It's like eating something and then digesting it down. But sometimes lysosomes can also be used to recycle a cell's own organelles and macromolecules, and this is called autophagy. So on the left is phagocytosis. This cell took in something and it just like pinches in the membrane and then forms a vesicle called a food vacuole. And then the digestive enzymes inside the lysosome, after the lysosome fuses with it, will like come in and break down whatever that is in the food vacuole. And then on the right is an example of autophagy. So say in one cell, something happened and some kind of like organelle gets damaged. So the cell will close that organelle that's damaged into a vesicle. And then lysosomes, again, will fuse with them and start breaking them down and reuse the pieces. And so that's called autophagy. So kind of like self-eating. And so we just talked about food vacuoles. So vacuoles are large vesicles, and they're derived from the ER and the Golgi apparatus. And they actually do a variety of functions in different kinds of cells. So that's why it's, diverse maintenance compartments. So those food vacuoles are formed by phagocytosis. There are contractile vacuoles in many freshwater protists. So again, remember, protists are like eukaryotic. They have membrane-bound organelles, but they're single cellular. And so many of them have contractile vacuoles that pump out excess water of the cells so they don't like swell up from being in a freshwater environment. In plant cells, there might be central vacuoles. So this is found in many mature plant cells, and they contain a solution called sap. It's the plant cell's main repository or like place to store inorganic ions, including potassium and chloride. And the central vacuole also plays a major role in the growth of plant cells. So here is a picture of a plant cell generic. And so in a plant cell, you will see a huge central vacuole to kind of store all these ions and sap. And again, not all the cells are going to look perfect as we kind of try to show them. You might see cells that are brick shaped, but some that are not so perfectly like brick shaped. So yeah. So here's a review of the whole endomembrane system and the components and how they work together. So the nuclear envelope is connected to the rough ER, which is also continuous with the smooth ER. If something is made here, the membranes and proteins made by the ER will move via transport vesicles to the Golgi. The products might get modified, and then the Golgi pinches off transport vesicles and other vesicles that will maybe give rise to lysosomes or other specialized kind of vesicles and vacuoles. If, needed, the lysosome is available for fusion with another vesicle for digestion. And if this is a cell that has to make and then secrete something to other cells, then the transport vesicle carries proteins to the plasma membrane, and then it fuses with the plasma membrane and then secretes stuff from the cell by this process called exocytosis. So I think I'm gonna pause there.